And what if you put it in el collar?
Okay.
Ándale,
Clara,
sí.
is that better?
Maybe yes, or this. Ahorita puede. Un poquito más arriba. Ajá,
Like that?
yo creo que sí.
Or should I use?
Yes.
Yeah,
No,
is that better now? Okay, thank you very much. Okay. Well,
Thank you. And now is the time for Claudia Martínez. She's coming from IDT, and she will talk about microbial expansins and Mutualists fungal effectors. Thank you so much, Claudia.
for organizing this very interesting meeting. I've enjoyed it a lot, and well, I'm very, very happy for him and grateful. Okay, so today I'm going to tell you about what we have been doing in my lab for the last 10 years, I think. We decided to work with these proteins, expansins, and at the time it sounded fun because we thought that there were proteins that would help to produce bioethanol. That was the idea behind studying these proteins, but then we realized that that was not the case. And so we needed to go back and try to understand what they were doing and understand why they were not helping for what we wanted. And so this is a long story, sometimes frustrating, but it's been a success on the way. So there we go. Okay. This presentation is going to be divided in three parts. First one and third are going to be very fast. I think I'm going to spend most time in this second part. And I want to tell you first about effectors and hypersensitive response, because I think in IBT, in general in this country, we don't, from these plant-microbe interactions, we don't focus most on that, not a lot on this part. And it's very interesting. I have learned about this only recently. I'm not a plant scientist, but I am interested in this response, and you will see why later on. Okay. So to establish a successful infection, microorganisms produce effectors. And effectors are molecules that are produced by the microorganisms, but that have a different target, have a target in a different species, in this case, plant. And so to be able to colonize, in this case, Pectobacterium is a bacteria that we work in the lab, and this photo was taken by one of my students, to be able to colonize and to invade the plant, the microorganisms have to manipulate the plant. The effectors have many different shapes on molecular nature, but I'm interested in protein effectors. Protein effectors are produced by the microorganisms. Normally, they have been studied in the pathogen, phytopathogens, but now we know that they are also produced by not pathogens, by mutualists and symbionts. Even this morning, I was reading about rhizobia. They have, some of them have type 3 secretion systems, and they sometimes activate immune responses in the plant. Okay. They target plant components, and these targets can be outside the plant, in the apoplast, or they can be intracellular. Those that are intracellular are directly inserted into the cytoplasm by specialized structures used by the microorganisms. So like fungi use Ostoria, bacteria use type 3 secretion systems, and nematodes, they have a needle-like structure that injects the cell, and with that they also introduce these effectors. So these effectors affect the whole physiology of the plant to be able to establish in the plant. So some of them also reduce or suppress the defense systems of the plants and will this for a productive infection. Okay, so this is only the structure of the different components used by different pathogens, or not only pathogens, also mutualists, to inject these effectors in the cytoplasm, the type 3 secretion system, Austoria, stylet. Okay, so this people refer to in the literature is that there is a co-evolution, because we have molecules of the microorganisms that have targets in another in plant in this case, and so both of them co-evolve. So if the pathogen or the microorganism does something, the plant responds. I don't think you can hear me anymore, or they have reduced the volume. Okay, so there is a host protein that is targeted by an effector, and so through the evolution, this results in plant manipulation. During evolution, through time, the plant then evolves a strategy to contend with that manipulation by the microorganism. And typically, or what I've learned more about, is the NLR proteins that they respond to. They have evolved to identify the manipulation by the effector on the target. And so these proteins that can also be, I'll tell you in a minute. These either the complex or the effector itself can be sensed by these receptors in the plant, or there's very nice examples in which the target protein of the plant is inserted into the structure of the receptor, and then when the effector targets that, then this activates the response. And this results in the hypersensitive response that is based on these proteins, and this hypersensitive response kills the cells and prevents the propagation of the pathogen. And therefore, as I was saying before, there is a massive expansion of these genes, either receptors in the surface or intracellular receptors, because they have to keep evolving to be able to recognize all the different effector types in or the microorganisms produce. So this is just the phylogenetic diversity of NLRs of plants. And the mechanism of action was recently discovered. It's very straightforward, very beautiful, because what happens is that these receptors exist, there's still, they are a diversity of them, and they act in different ways. But some of them are monomers or sometimes dimers, and they recognize when they interact very specifically with in this case, a protein that is part of the plant that has been modified by pathogen protein, and then this promotes the oligomerization of this structure, and that is called a resistosome. And this then inserts into the membrane and produces a sudden calcium entry into the cytoplasm, and this results in cell death. Obviously, this is not the only mechanism, but it's beautiful, right? How this evolved for the plant to have a very fast response when it encounters a pathogen, right? Okay. So this cell death, let's call it cell death. This cell death, what it does is kills cells, and it prevents the progression of the infection. And Nicotiana is an excellent plant model for reconstitute interactors, so effectors, targets, and receptors, to induce the cell death. And this is a very nice system that we have in the lab to use. Okay, so, and this is very important because finding these interactions can help developing pathogen-resistant crops. When we know how these interact and that this is going to give us a response that is going to stop the pathogen to keep progressing, then we have a powerful tool to try to induce resistance in crops. Okay, so that's just like an introduction. Now I'm going to tell you about microbial expansins because we think they are effectors. Well, they are in the broad definition of the effector. So we've been working with these expansins, and as I told you, we were working with them for completely unrelated reasons to the interaction with the plant. They exist, or they were first identified in different plants. They act non-catalytically. They are not hydrolytic enzymes, but they lose the cell wall. They interact with different components, with different polysaccharides, and they induce the relaxation of the cell wall. In plants, they exist all over. They have important roles in physiology and in processes where cell wall needs modification. Growth, fruit maturation, the stress responses, they all involve expansins. But microorganisms also contain expansins, and their function is not understood. So that's what we want to know. Structurally, expansins are small proteins. They are composed of around 210, 220 amino acids, and they are divided into domains, domain 1, which is down here, and domain 2. They form a compact structure, and among them, there is a flat surface where a polysaccharide can be bound, and this surface is composed of, how do you call it? Conserved residues. Microbial expansins have the same structure, but they are very divergent. In different microorganisms, their sequencing identities are very low, but structurally, they're very similar with the conserved residues all in the same place, independently if they are plant expansins or microbial expansins. Okay. In plants, a canonical expansin is formed of these two domains, although you can find individual domains. These are the active forms that have been reported and that are considered expansins. These are not. In bacterial genomes, we also have the two domains, but sometimes we can find them also associated to other catalytic domains. And also we can find them just this domain 1. In eukaryotes, like fungus, we also have the canonical structure, but also associated with other domains. And interestingly, lots of effectors have been reported to be single domain 1 expansins in fungus. And in nematodes, this activity seems to have evolved convergently because DPB domain, this domain is homologous to all the others, but domain 2 is not is not a carbohydrate binding domain, family 63, like I didn't say that, but it's a binding domain. In this case, it belongs to family 2. And also the architecture is the other way around. So the DPBB, the domain 1, is on the C-terminus instead of N-terminus. But the activity seems to be the same, seems to be also conserved. Okay, so we wanted to know how many bacteria had expansins. And what we found is that they are not very prevalent. They are actually very rare in bacteria. If we analyze using this anode tree site, which contains more than 3,000 bacterial species containing more than 140,000 genomes. And we ask for something similar to Expansin from Bacillus subtilis, what we get is only 638 hits. And if we consider that bacteria have only one gene, what we have is 638 only genes in all these databases. So what this tells us is that they are very rare. Interestingly, they are along all the different bacteria types. They are more concentrated in some type of bacteria, but they belong more or less everywhere. So Andreas de Sandosecchi, he is a postdoc here, but he was a PhD student in my lab. He decided we wanted to understand more about the phylogeny of these proteins. So he got, with bioinformatics, 3 other genes, and then we compare them, and we can classify them like this. So they belong mainly to Firmicutes and Proteobacteria, and Actinobacteria. They also have quite a lot of species that contain expansins. As I said before, they can be associated to other domains, but what we know, well, this is again a work of Andres, and this red is the compact form of the 2 domains, and this is just to show you how different they can be. But we just focusing on the canonical domain, what we see is that they are very highly charged proteins, either positively or negatively. But what they have all in common is an electronegative pseudocatalytic pocket in the same site. And we think this is important for activity, because if we mutate this site, this residue, which is an aspartic acid, in Bacillus subtilis is position 82 or in Brasiliensis 83, then we abolish the activity. So that's important. What the electrically charged residues are doing in the surface of the proteins, we still don't know. Okay. What we know is, is that these all, as I said, all effectors have to be secreted. Because the target is is outside the microorganism, they have to be secreted. So that's one of the things that we always look for when we try to analyze effectors. If this is not secreted, then it's not an effector. Okay, so they are all secreted, and what we saw is that these expansions would just go away, like solubilize in the medium. What we found through Andres' work is that they are associated. Well, the sequences tell us that most of them, not most of them, half of them are associated to the cell exterior, to the bacteria exterior. And that tells us that then maybe the expansions are also doing something in bacteria. That's a story I'm not going to tell you today. I don't have time. We created a mutant of expansin in Pectobacterium atrosepticum, and that doesn't give us any phenotype. The only phenotype that we get is, it gives us 2 phenotypes. I'm lying. This one, which is the swarming, the movement of the cells collectively in the surface, and expansin mutants. This expansin mutant is unable to do the swarming. I'll show you later. So we have this phenotype that is associated independently with of the bacteria interacting with the plant. So Gloria is analyzing the role of expansin during the swarming of Pectobacterium, not only atrosepticum, but also Brasiliense. She's presenting poster number 31. And also what we want to know is what does swarming have to do with infection, because it's not reported for this bacteria. And we think that expansin might be related there, but we're still analyzing that. Okay. We also know that expansins, as I told you before, are present in other organisms. In here, I show you this number of genes that are retrieved from the database searches, and mainly we have found them in fungi, but we also find them in nematodes. And this is very interesting because although we see a variety of microorganisms that contain expansins, if we analyze this table where we rank the top 10 plant pathogens, either bacteria, fungi, or mycetes or nematodes, all of these are the ones in blue contain expansin genes. And so what this is telling us is that they are doing something in the infection, right? What they're doing, we still don't know, but it's very evident, especially for bacteria. If you remember, I told you that bacteria they are not very prevalent in bacteria, but it's very interesting that those that are very bad pathogens, they have expansins. Okay. So what people have done is they have mutated some expansins. There's not much in the literature. They are not easy proteins to work with. And what we know is that Bacillus, Ralstonia, and all these pathogens have, most of them, reduced symptoms. So this is the one we created. As you can see here, the wild type Pectobacterium, that Atrosepticum, that only produces symptoms in potato, have these lesions, and the mutant is reduced in the symptom. Also, this other one that infects squash. Yeah. So we wanted to know, what about the electric charge? So we produce in the lab Expansin XL1 from Pectobacterium brasiliense and XL-X1 from Bacillus, and we use them because both of them have different electric charge in the surface. And so we isolated cell walls from different plants, and we put them to interact with the expansins. And what we see is like a mirror of interaction. So like where Bacillus expansin binds more, Pectobacterium binds less. Yeah, and we see this, and we think that it has to do with the charge of these proteins. And so we wanted to see where they were binding in the tissue. So we fluorophore mark both expansins with site 3 and put them to interact with, in this case, this is celery. We have done it with other tissues, and we have the same results. Bacillus subtilis binds everywhere, whereas Pectobacterium expansin binds only in the vascular system. And we can interpret this result because Bacillus expansin is very positively charged, and it interacts electrostatically with the negatively residues of the cell wall, whereas Pectobacterium expansin is negative, and it only bound here, which was very surprising to us. So we know that apart from the polysaccharide binding surface, electrostatics also have a role. Then we compare the two types of binding. So this is Bacillus, it binds everywhere, and this is Pectobacterium. And what we see is that because we can see all the cells marked in this photograph, we realize that around the cell vessel, the xylem vessels, there's surrounding cells, these vessels. And what we saw is that the Pectobacterium doesn't give us a homogeneous binding. It's like rotate. So when we zoom in there, what we realize is that the expansin is binding to the cell corners of the xylem vessels and the surrounding cells. So Mary, she was a master student in the lab. She tested different antibodies directed to different polysaccharides, either hemicellulose, pectin, or cellulose, to see if any of them would give us a pattern similar to what we see with the expansin, in trying to understand what is in that area that might be a target for the protein. So she did that, and many of them didn't give us a a pattern that was similar to this, but we found these were the most similar. So then we thought, okay, well, we have maybe these three different components. They are all type of pectins. And so next question is, which one of them is the one that is the target for the Expansin? So what Mary did is she extracted pectin from the vascular vessels of the celery, and she produced 3 different extracts with proportions, varied proportions of the different components that she determined using these antibodies in like ELISA type experiments. And we also use a pectin that is commercially available. And that's this is the different components, the different proportions of the components that we have. Then she covered the walls with the polysaccharide, with H extract, and then added the protein, and we look for what was giving us signal. And so we got this result, and we found this amount of protein bound to different, so each extract. And so comparing the order of binding of the Expansin to the different extracts, what we see is that this component here, the ferulylated beta-D-galactan, is the predominant component that we think might be bound by the Expansin. And this is very interesting because previously we have already reported that if we incubate I forgot ¿cómo se llama, Juan? The maize hairs, how they call? El stigma. So maize stigma, cell walls, because they have been reported that SEA maize, XPB1, releases something from maize stigma. So we isolated the cell walls from that substrate and incubated with XL1, and we also saw that something was released. We don't know why, but this is a component that absorbs in the range of I forgot the name. Like ferulic acid and caffeic acid, all these acids that have a what do you call it, Benson ring. So that was like very interesting to find out, but we still need to confirm this. So we have many open questions for the role of XL1 in the plant infection. So why is it? Is this protein important for xylem colonization? We don't know. Is vascular pectin a determinant for niche detection? Because Pectobacterium moves through the xylem vessel, so that might be one possibility. And so we have tried different microscopic techniques in trying to see the movement of wild-type cells of mutant Expansin cells. And so we have this very beautiful system. Sadly, we did that in Arabidopsis roots, and Arabidopsis are very tiny. And so we were inoculating the bacteria with a needle by hand, and sometimes we would put the bacteria right in the surface, and some other times we would cross the root, and sometimes we would put them directly in the xylem, and that was not useful. So we've been trying to optimize this technique. But Mari was trying many different things. And here I'm going to open a parenthesis because I'm going to tell you something I wasn't going to tell you, but Jose said to me yesterday, no, please show it. And then it's interesting because Nazar also showed it. And it is that trying to have a system of interaction of Pectobacterium with Arabidopsis, Mary did many different things. And one of them was with Salvador, and she established this co-culture system of Arabidopsis, and she placed the bacteria inoculum in different places of the plate cell, trying to see which one would give us a way where we could see the colonization. When we did that, we realized that Pectobacterium doesn't grow in this medium. It only grows when the plants are there. So that's very interesting. And also, what she did lots of measurements. Because Pectobacterium is a pathogen, it's a necrotroph, it's a very aggressive pathogen. If we put them at some distance of the plants, the length of the main root is always very affected. Leaves are okay, and in general, the plants are not very healthy, but they are okay. They are more or less. But what we saw is this: whenever she had the bacteria here, the roots were bending to the left, and we thought, oh, that's very cool. So the bacteria are calling the roots, right? Because they want a plant to grow, they want a plant to infect. So they are calling the roots. But then Arturo suggested, okay, well, to see if this is real, before we even did a very nice correlation of the angle of the roots with the distance, and it's perfect. We were very happy. We thought, okay, well, this is a very cool phenotype. But when we put the inoculum in the other side, what we saw is the roots are still going to the left, right? And this is our wild type Arabidopsis. We didn't follow this because we thought, okay, well, the bacteria are doing something, but we don't know what. And because this has more to do with the physiology of the plant, and we are not plant physiologists, we left it there. But now I see that this phenotype is everywhere. And that's why I wanted to show it. We would like to know why this is happening. I guess this is a question we all have. This is where I'm going to close the parentheses, and I'm going to keep going with the expansins. So I was telling you about the effectors because we came across this plant immunity when we were trying to find a phenotype that we could measure the activity of our expansin with. And so what Delia did for her PhD is that she infiltrated the pure protein in Arabidopsis leaves, and 24 hours later, we challenged the infiltrated leaf and look for the and then challenge with the bacteria and look for the phenotype. And what we noticed is that infiltrated expansin protected the infection of the bacteria, whereas the non-inoculated very susceptible. And so that's why we came with Mario, and he helped us to understand what's going on here. What we see is that if we just infiltrate the leaves, we have this increase in weight loss. So this is like counterintuitive interpretation. But when we protect the plants infiltrating the protein, we have less maceration. And this is dependent on the residues that I told you were important for the activity of the protein. If we mutate those residues, we don't see the protection. We saw this for some time, and up to 72 hours. There's no significant difference here, but we still see a little difference. We did a dose response curve. The more protein we add, the more protection we have. And not only it protected against Brasiliensis, but also protected against botrytis. And that was very interesting. Then Delia analyzed different genes and did lots of work with this. We concluded that immunity seems to be triggered by the activity of the protein, not by the protein itself, because if it was the protein, then the mutants would also protect, and we don't see that. So it might be that the protein displaces a molecule, a polysaccharide, during its activity of loosening the wall, or it could change the mechanical properties of the cell wall. She also showed that the immunity depends on jasmonate and salicylic acid and ethylene. We did also a transcriptome that we're still analyzing. We're still working in there, but I was thinking with your talk, Cyril, that maybe we can look for the scoops in there, because you said that they are induced when there's elicitors, so we have an important elicitor here. Okay. So when we realized that these expansins were triggering immunity, then we wanted to know if there was anything else reported. And what we saw is that XPV2 from Globodera is a pathogen nematode that produces these cysts. This is XPV2. It's called an Expansin, although it's just made of domain 1. It triggers cell death, right? And if you remember, cell death is a protection system of the plant. So we ask if this could be also the case with our Expansin. So we express Expansin XL1 from Brasiliense in Bentamiana. And luckily we have not a very strong response, but still a response. And again, this depends on the amino acids that are important for the activity, right? So those are the amino acids. And so please go to poster 30 because Carla has more details on this experiment. But we want to know now what's the reason for the cell death phenotype, how it is induced, and what's the stimulus that it's been detected. So we are answering that with Carla's project. Talking about nematodes, there are two types of phytopathogenic nematodes, and they are very important in the agriculture because they cause great losses. Okay. The two types of nematodes are the sedentary nematodes and the migratory, how do you say? Migratory nematodes. Okay, so interestingly, the two families, the two genera that are very important in agriculture is Heterodera and Globodera, and then the Meloidogyne. These are sedentary nematodes. If we analyze the expansins, they are the two-domain expansins and the single domain. The migratory nematodes, they only have the single domains, and they seem to have acquired it independently. Sure enough, we look for how they cluster with other expansins, and the nematodes are here, very close to the bacteria. Is it here? This one, I think. And many of the enzymes that degrade the cell wall from nematodes have been obtained from bacteria. And so, what's his name? Sebastian Van der Acker from Cambridge University, he gave us the transcriptomic analysis of what they did is they extracted the RNA to sequence from every stage of the life cycle of Heterodera's cacti, and he gave us all the data, and we analyzed the expression of 3 different expansins. As you can see, the nematode expansins are completely different in architecture and also in structure. Remember that I told you that expansins are compact proteins. In nematodes, they are like open structure proteins. And what we see is that they are produced, these 3 different expansins that are encoded, that are expressed from this organism, are mainly being expressed in J2, which is this phase before the nematode enters the root, and 10 hours later, after it selects a site where it's going to remain the rest of its life, the expansins are more or less repressed. So that tells us that these genes, at least here, are expressed during the moral phases of the nematodes. We have established a population of Meloidogyne enterolobi in our lab, which hasn't been easy. These are parasitic, obligate parasites. We need to keep producing them in the plants, and it's not easy, but we have them, and we're going to analyze of the 54 expansins that have been predicted in the genome of this nematode. 3 of them only are composed of 2 domains. The other 51 are only 1 domain. But interestingly, this here I put the comparison of these 2 domain expansins, and very divergent. And they are very divergent because they have the two domains that I have been talking to you about connected by a very long unstructured sequence that we don't know what they exist for. So we predicted the disorder of these proteins, and they are disorder proteins. And so we decided to analyze if these disorder regions can acquire structure, because these disordered regions acquire structure when there is a stimulus, right? And so there has been reported a biosensor to determine the structure acquisition by IDRs, or these intrinsically disordered proteins, and it works like that. So we can clone disorder region between two different fluorophores. In this specific biosensor was designed to determine the acquisition of a structure in a very crowded condition, and we can create that by just adding salt into the system. So by adding salt, the cytoplasm becomes very crowded, and that is the stimulus for this region here to get a structure. If that happens, then the fluorophores get together and we can have red. So that biosensor has been reported, and so we clone our regions among these two fluorophores, and we tested for the acquisition of protein. The more red, the more energy transfer from this fluorophore to this other one, and this is the one that we detect, then the more this is closer. The closer they are, the fluorophores, and that means the structure is more compact. Okay, so we did that, and this is our positive control. We see FRET using this Z1, that is a Lea protein from, I don't know where from. Is it from, from Alejandra's lab? Frijol, okay, bin. And so we tested our proteins. One of them doesn't seem to respond, which is cool. We can compare and see why it cannot be acquiring structure, but the other two respond even better than this one. No, they respond faster, and this is increasing the salt concentration, and they respond better. But clearly, we don't know, or we think that in the apoplast of the cell we will find high concentrations of salt. So what could be happening in the apoplast that could be triggering this response? Well, one possibility is a change in pH, or maybe the oxidative environment that is produced by the plant when it detects that something is not there. So now we are going to test these regions with different pH and oxidative stress, both in this system that we have that is based in yeast, but also we are clone them already for expressing them in Bentamiana, and see if we can see FRET, maybe see if we can find condensates in the apoplast that have never been reported. Maybe not, maybe this is just a crazy dream. We will see. This is the work that Carla is doing along with Lilian. So again, if you want to know more about this, please go to poster 30. And so, in conclusion, microbial expansins and expansin-related proteins that we call the nematode expansins have a role in the interaction with microbes and their hosts. And they have evolved convergently in different phytopathogens. Expansin is important in this particular one, Expansin 1, is important for at least for the swarming phenotype in Pectobacterium. This same protein seems to bind a pectin ligand in the vasculature of the celery and other species. We don't know if this could be important for silent colonization. It triggers plant immunity. And Meloidogania interlobi has two domain expansions related to related proteins that have different architecture, and they might have evolved to contend with different conditions. We don't know. We are testing that. Okay, so changing gears again, I'm going to tell you something we have been doing very recently. Why is it turned on? I see your face there. Oh, no, this is quick. We have been doing, we decided to also not just work with expansions, because as I said, they are not very easily proteins to work with. But then we are also working with two mutualists, and that has been the work brought into the lab by Iran Tapia. She's a postdoc in the lab, and we did this because during our experiments with the nematodes, we identified a strain that was killing them, no? A fungal strain that was killing the nematodes, and we thought, well, that's very cool. Let's isolate that strain. So we isolated. This is the mycelium growing on the eggs of the Meloidogania interlobi, and we isolated it into an axenic culture, and this is the reproductive structures under the microscope. When we discovered this fungus, we were a bit disappointed because this fungus is already being used as a control agent for nematodes. So we're like, oh, okay, well, it's been described. But interestingly, there's not much in the literature. So we have a lot to learn from that. We confirmed that it's indeed killing the nematodes. And it is an Ascomycetes filamentous fungus. As I said, it's used in commercial compounds. And that is very well represented by Juan, and that Botarga 2 weeks ago in the Carrera de Botargas. And it was very interesting to learn that under stress, plants under stress, in this specific case, tomato, become susceptible to the infection of purpuricillium, and it becomes an endophyte, and that's been very interesting. In this case, we identified cold stress, but we have tried other different stresses, and again, it endophytes the tomatoes. So that's something that we are studying now. Go to poster 5. So there is a big effort to identify effectors of pathogenic fungi. But also, they can serve us to identify effectors in mutualistic fungi, right? So we have applied this pipeline. It's a bioinformatics pipeline that detects these secreted proteins. They have to have certain characteristics, and if they have them, it gives us a score, and it tells us if it is an effector or not. So we have found quite a few effectors in purpuricillium, but some of them have similar sequences that have been already reported in pathogens, and these are some of them. These are the structures, and right now we're trying to understand if they are produced by purpuricillium and if they indeed help the infection. Second mutualist is Naganitia adelense that you might have seen in poster number 6 by Iran. She isolated this. It's a yeast, Bacillomycetes yeast, from a high-altitude mountain in Estado de Mexico. We have sequenced its genome. Here I show you complete chromosomes. As far as we can tell, it contains 17 chromosomes, which is very cool. It has so many different properties that we see in the genome. One of them, I put it there for you, terpene synthesis. But we also, I mean, this is not the very highest quality picture, sorry, but we are, because this is very recently, Iran saw this yeast inside the roots of maize, and they seem to be also endophyte. So that's also very nice to see. We're going to characterize this in detail. But this is what happens. Let me show you. Iran wants to test if this Naganitia can do something against nematodes, because we know that these yeast promote growth. They don't. They don't reduce the number of nematodes. They don't reduce the Gallin index, which is how we see the infected roots. But what it does, it makes the plant blind to the nematodes. So the plant grows in the same way as the non-infected plants with nematodes. So this is a control plant that hasn't got the yeast infected with the nematode, but this is the one that contains the yeast and the nematode, and the plants are the same as the wild type. So this is very, very good. Because we think it's an endophyte, sure enough, we also found some predicted effectors. And so what we want to do is to understand how these two mutualists colonize their host. We're doing transcriptomics to identify pathways in which they can interact with the plant. We have described, we want to describe the phenotypes associated with the endophytism of these two different strains. We have to confirm that, but mainly what we want to do is confirm that what we see in the lab can be translated into the field. And for that, so this is, for instance, plants that have been treated with purpurylicium, and we think that these have been endophyted, if we can use it as a verb, and these haven't. So for that, we have a collaboration with people in here in this place in Tlacotepec, in Zacualpan de Amilpas, which is in the eastern of the state, and we're going to do experiments at big scale with first with Naganitia and then with purpurylicium, because what we want is to see if really these things work. No, that's what we will all love. And with this, I want to just show you who we are right now. This is also from 2 weeks ago. We were uniformed, and I want to thank Daniel because he created a beautiful logo for our group. And this is this is our current group. All the people that have been in the lab are here. More or less, I show you who they were in the presentation, and these are people who have been helping us in our collaborators. And thank you very much for the sponsors, and thank you. Yeah.
Thank you for the talk. I have a question about, can you hear me? I have a question about the part where you said that you identified expansins in bacteria, but that you found way less than you were expecting.
Well, no, we weren't expecting anything, but what we know is that there are only a few hits.
Okay, but then what I got from your slide was that you just took the Bacillus subtilis expansin one and tried to see if it was present in all these genomes that you as a theory. But you mentioned that and you showed that there's so much diversity in expansins, right? So this number could be severely underestimated because maybe other bacteria have expansins, they're just not as similar to the Bacillus subtilis one.
Yeah,
so I have a suggestion. So, have you thought about every time you identify one that you consider a hit, then you start over on all the genomes from that new hit, so that you recover all the ones that are more similar to this new hit instead of Bacillus subtilis, and that might expand considerably your yourself.
Yeah, we haven't done that. That's a good idea. Yeah, we'll try it.
If you send me an email, we can.
Yeah, I will. Don't worry.
Thank you.
Thank you. Yeah.
Sorry?
No, rhizobia hasn't got expansion. No. But it has T3SS system and it produces effectors. That's something I learned but not expansins. No.
Thank you. Yeah. So I want to know, like, for instance, expansin are very sensitive to pH. So do you know if all these expansive that you have been analyzing is pH responsive?
They don't seem to be very sensitive, but they depend on the pH to be active. So what we have done is done all our work with Expansin from Lactobacterium at pH 7.2.3 because we measure the pH of the infection. You know, that's very easy to do because you infect the tissue with the bacteria and it macerates everything. So you can recover the supernatant very easily. And we measure the pH there and it's around slightly alkaline. And it's been reported that these hydrolytic enzymes of Lactobacterium work to that in that pH more or less. So that's the case for this one.
Yeah, because pH around the cell wall, it's much more acidic than that, isn't it?
The pH in the root?
Yeah,
I don't
cell
know.
wall, in the cell
I don't
wall
know.
in general. Yeah, probably that's another comment that I wanted to tell you about the sensors, the way it's working. That I mean, it depends also the GFP that you are using. That may be very pH sensitive. I mean, it can work easily in the cytoplasm, but it might not work in the same way.
Yes, completely. No, we need to think of all the different controls to confirm that what we see is real. Yeah.
And then my last question is related to the binding of the expansin to the pectins. Do you know is binding the same way to the esterified pectin or the esterified pectin? Is there a difference?
Mary did that. If you see, sorry, I think it's important that you see it. Here. Here we have an antibody against methyl esterified pectin and non-methyl esterified. And we didn't find a match, let's say. Let's call it like that. We didn't. These are the ones, these other ones. So this is the fair related, the Cylogalacturonan. Oh no, yes, sorry. No, this one. But this, I'm not so sure because this one, although this is a bit more different, but yeah, so that one is. I was saying something.
Thank you.
So thank you so much, Claudia, and see you at 4 p.m.
Thank you.
She's coming from IDT, and she will give the talk about something with, but yes, the next something about mitosis, right? Or some cell division. Just okay. Thank you, Rosana, for coming.
So I would like to start and repeat my name. I'm Rosana Sánchez. I work in the IBT next door. Belong to the Department of Molecular Biology, Plant Molecular Biology. So I would like to start by repeating, insisting what it was being here yesterday, that the idea of having this symposium, it has been a splendid idea. I am very glad to meet you and to have the opportunity to talk about science in a very relaxed way. So I hope in the next 2 years, we'll have the same experience and repeat it frequently. So the title, as you can see, refers to cell division. Jesus insisted that Phaseolus vulgaris is not a good model to study. And I will say that it's a very difficult model for several reasons. It is a very good model if you are wanting to address very specific questions, especially in the dilation. So I will say that I got my PhD back 1989, working in bacteria and animal cells project. Then I switched and I started to do molecular biology in parasites, in protozoa, mainly malaria parasite and amoeba. As a Mexican, I have to understand what's a parasite in Mexico. And for the last more or less 15, 20 years, I have been working in plant biology, but I will say that I consider myself, considering all my transitions in the field, I consider myself a molecular and cellular biologist. And I was convinced that textbooks now are really oriented for young people to understand what is known now in molecular biology or cellular biology. But in some way, they are also misleading the concepts because they summarize what is known in very well-studied models. E. coli is bacteria, and all bacteria, they have behaved the same way. And we have, Saccharomyces cerevisiae, Drosophila, mouse, zebrafish, and Arabidopsis thaliana. So we assume that what's been published, what is in the textbook, is going to be repeated independently of which species or kind of organism we are studying. We have heard here that it is a repeat concept that comes to us, that our model has some peculiarities. So I'm interested in Phaseolus vulgaris because when I started to work in plant biology, I'm not a plant biologist, for sure. Don't ask me physiology because I'm not a plant biologist, but I am a molecular and cellular biologist. So I consider that the nodulation process is quite rich to understand several events that has to be done in a very well-coordinated way in order to have a succeed nodule and happy fixing nitrogen. So why I do study or I use Phaseolus vulgaris as a model, it is because I'm really interested in cell division. I always said in my life that I would never go to the cell cycle. Forbidden for me working in transcription factors. But I reflect about that. So let me say that I'm studying that particular model from a molecular and cellular perspective. So I first thank my team because they are hard workers and they are interested and they are always ready to say whether I do agree or disagree of what I am telling. So I really appreciate that. So we have here Eric. Here, he is a postdoctoral. He's the plant biologist in the group. So all the plant biology questions, address them to him. So I do have now two very nice, kind students, Livni and Mariana, that are present here. So they are doing their master degrees working with Phaseolus and trying to deal with the model. Former students are Raul, who was working with CMRK that we mentioned early in the morning. Here you have Elizabeth Monroy, Ellie, mentioned by Jesus. She did a PhD in my group, working with a very specific question that is responsible for all my present interests. And I will summarize a little bit her project. She's really hard worker, very kind person. And I am glad that she is happy working with Jesus. Here you have a Catalina, also mentioned by Jesus. She did a master's degree in the group, and I'm going to present part of her results. She's very kind, hard worker as well, and extremely friendly. So thanks to both of them for all the work that they have done in the lab. And there is a list of former students, including Elizabeth, Raul, and Catalina, and 3 bachelor students that they have been quite useful for us in the last 4 or 5 years. Mariana is here. She was trained in for the bachelor degree in Jesus lab. So we switch students now, and she is living working in the lab. So I hope that both of them, they will present the results soon, at least in Merida. So we have here the model that we are using, legume rhizobia interaction, because it's a very challenging model. It does things that it is not obvious that a plant is going to do. So I'm really interested. And talking about different models, we have Medicago truncatula and Lotus japonicus. Already Jesus mentioned a lot of very nice qualities in Lotus japonicus. But I would say that eventually they are competing in terms of kind of research that people is doing in between researchers using Medicago truncatula as a model or Lotus japonicus. Both are quite interesting. Glycina max soybean is another quite interesting model. I don't know how difficult it is to work with, but we have Phaseolus vulgaris, our study model, which becomes infected, and we use Rhizobium ethli as a rhizobia strain. Jesus already mentioned there are at least 2 large groups of nodules that are this kind of shape, like ovals, and they have a very peculiar characteristic, like the apex here. It is keeping the proliferation activity in the nodule, okay? Whereas in the nodules that are produced in all these 3 legumes are nodules that are round and considered determinate because proliferation stops when it is the right moment. So with these characteristics, we can address different questions. So let's. So Jesus and Gina and Esperanza have already summarized more or less what is a nodulation. And it's a quite interesting process that, according to the data that is now, it's a negotiation or a convincing dialogue between legume and a bacteria, rhizobia. So it is true that it took years and years and years. As Jesus mentioned, it's highly probable that they started by this intercellular invasion and then realized that it could be also quite important to use in a more specific way to get, or successful way to get into the root. So with this negotiation, the bacteria, first of all, had to convince the root, the legume root, that it was a kind friend, and that the bacteria was not pathogenic, first of all. Second, the root has to adapt their physiology, genetics, biochemistry, biology to create a new organ. And it took advantage of what is known in terms of how a lateral root is developed. And nowadays, we know that a lot of genes that are involved in forming a lateral root in the legume root were just adapted to start to generate a nodule. So with all of this information, now we have just an immense amount of knowledge which genes are involved in starting a nodule, in preserving a nodule, and invading a nodule, et cetera, et cetera. And all of these genes have been characterized in terms of sequence, whether there are conserved genes coding for other proteins in other organisms. But it is a very particular thing that it is always quite of challenge for me. Because whenever you read a paper in which they try to characterize a mutant, it is true that in general, people that is working in nodulation focus on the phenotype of what is the problem in developing the nodule. And not, or a few of them, they also describe what is the real phenotype in the root. How many lateral roots they have, how long is the root, how well it's functioning, how many layers has, et cetera, et cetera. So I think it's just a problem in our knowledge, because we used to call nodule-specific genes, but you realize that the gene is there and it is expressed, and nobody cares or is interested or really interested, or probably it's a difficult question, what's this doing for, like, you know, the normal biology of the root. Anyway, so Jesus already summarized that the root hair is quite important here. We have the rhizosphere here expressing this greenish circle. We have the flavonoids that interacts with the rhizobia, and in response, you have the node factors. At Zoom, the root hair responds to that particular molecules. And then they start the real interaction. So if you add to that system rhizobia, can you start the movie? It is important to have this movie. It doesn't introduce things that are not known, but I think it's to understand the question that we are addressing. It is important to know that in general, the root hair is just like moving in a way that they try to have this particular probe there in which the bacteria gets inside and provides the environment that rhizobia really likes and start to divide. So we say that after that, we do have a colony here. So when a colony is formed there, this interaction or talk between the root and the bacteria indicates that somehow part of the membrane that is here, sealing the yellowish infection camera, tries to be modified. Some information is available, but still it's like just a challenge to understand in terms of cell biology how a membrane gets invaginated and introduces. It is a way in which the bacteria gets into the root hair. So it's yellow because it's an extracellular, it's an apoplastic environment, which is outside, but inside, okay? So there is communication in between of them. All these blue dots that you see here, it's because it has been demonstrated that all these vesicle interaction, endocytosis and exocytosis, it's quite important for that particular function. New membrane is built, new cell wall is built, so makes sense. So after that, so it is well accepted now that the process of having this infection chamber and the construction of this invagination, which is called infection thread, and we in general call this process like epidermal infection, that the entry by the intercellular way that Jesus explained us, it's an epidermal infection, but this is mediated by an infection thread or IT. When this is happening, it's well accepted that somehow there is a program that's turned on, biological problem. A lot of genes have been identified, involved in this mechanism. But really, what's the mechanism is not very well described. So when that starts concomitantly, meaning at the same time, more or less, the cells that are cortical, at the cortex of this specific infection site in the root, start to divide. So what is the instruction like saying you have to divide? When you have to divide, how slowly you are going to divide is an open question. Then, I mean, the cells still continues to divide, and they form a primordium here, some mass of cells there, by definition. Proliferation eventually stops or are organized to produce a nodule. So by the end, we have a nodule like this, which is the nodule that we used to have in Phaseolus vulgaris roots, also fixing nitrogen. But we are particularly interested in these initial cell divisions that's supposed to be or are coordinated with what's going on with the infection thread. So it is a dynamics that has to be done, but it's a dynamics that we cannot describe in terms of time, because it's a long process, it's a continuous process, meaning that when you have a rhizobia that infects a particular root hair, probably tomorrow or 2 days after, there is going to be a new root hair that's going to be infected. So there is a continuous there. So it's quite difficult to establish the time window. So we decided to use a description in terms of what's going on. So there, our main question is, what is the dynamics between the IT formation and the initial cell divisions? It looks like a very easy question. It's quite easy. Let's see the division and let's see what's happening with the IT. So that's the strategy that we apply. And we say just this very simple thought will eventually lead us to answer questions. So we have here a scheme showing the root anatomy in Arabidopsis thaliana. And I insist that you have to know that, because that's particularly quite simple as a root. You have the epidermis in blue, you have only one cortical cell layer in brown, and then you have the vascular tissue. And you have different sections of the root, but particularly when you start to see these root hairs is because that part of the root is differentiated, and that are specialized cells. So all these specialized cells are interacting with the rhizobia, and in this scheme, summarize what's going on, or at least in terms of biology or cell biology description, what's going on. We have a huge vacuole because plant cells, they have huge vacuoles. I don't know why, but I mean, I do know. So you have here the formation of the microcolony or the infection chamber. Several bacteria are here forming the microcolony. We do know, because the movie that you have seen, it's clear, it's serious. The nucleus migrates to the top of the apex of the cell. The plasma membrane is there, the cell wall is there, and a lot of vesicles. But which is really important, it's to focus that we have to think about that that's a particular membrane that is invaginated. Next step, you have here. So whenever the infection thread is being formed, this is the first, we call this like initial IT because it's starting. So the initial IT is kind of really, really tiny tail that you can observe here. And the vesicles are showing that here, the apex, we still have to work building new cell wall and plasma membrane. And the nucleus migrates. So it's well accepted that the nucleus or the nuclei in the root hairs, infected root hairs, are driving the infection towards the base of the root hair. Later on, these infection threads still growing. It can just be ramified. I mean, you can have like here like a root, but this is in a simple explanation. It has to get to reach the plasma membrane of the root hair. So let's say that it's reaching here the base of this particular cell, okay? So it is reaching here. Next step, the infection thread invades, enters the next cell layers. If you have it here in the indeterminate nodules, meaning Medicago truncatula, it has a particularity other than the shape, the oval shape of the nodule. And very interesting particularity is that if you have here the pericycle cells, meaning the frontier, and you have here the vascular here, this blue cell is the frontier, and you have the pericycle and then endodermis. So in Medicago truncatula, there is a very well coordination of cell division as well, as in Phaseolus vulgaris, and all nodules characterized nowadays. The difference is here is dividing. So these same cells in this kind of layer, I mean, this type of layer, it's where a lateral root starts to be formed. They are primed for that. So these cells that divide here share a particular program similar to what happens in the root hair in the lateral root formation. It also happens in Phaseolus vulgaris on Glycina max, but it doesn't happen immediately as in Phaseolus vulgaris, in Medicago truncatula. If we check what's happening in Phaseolus vulgaris, Lotus japonicus determinate nodules, is that as soon as the infection thread goes through the root hair towards the base of the cell, this layer of cells starts division. So that's the reason why I'm studying Phaseolus vulgaris, especially or particularly because Phaseolus vulgaris first divisions occurs in the cell layer C1, which we used to call the subepidermal cells because are under the epidermis. So we are interested in tracking how this infection thread elongates, reaches this layer, which is by definition dividing, and goes towards the rest of the cortical cell. So I am interested in that particular scenario, okay? So to address that question, we have different strategies that we have to implement in the lab to be sure that we could do it. Hairy roots generation has been established for more than 20 years, so we just know how to do it. And here is Catalina trying to show us how we prepare hairy roots. A Senko tool that we have developed for the visualization of the infection thread is a particular strain in which everybody more or less use it. It's expressing DS red, but since in general the DS red sequence or coding sequence is in a plasmid, you used to lose the plasmid. So we collaborate with a colleague that was doing his PhD here in the CCG, and he created that particular strain in which DS red is in the chromosomal DNA, so doesn't lose the signal, which was a problem, or is a problem with the conventional strains. So, I'm sorry. So just to show you, because it's our main tool, we just track the infection thread by appreciating how complex is the infection thread using confocal microscope, and we can just appreciate how it is growing or elongating or invading or getting more complex the infection thread. And here you can see that the infection thread is growing there. But the thing is that this field, it's not a fluorescent field. And Phaseolus vulgaris, one of the disadvantages is that it's a really thick root. So it is quite difficult to study using my confocal microscope. So since it was complicated, we decided to do analysis of different genes and starting, because we don't have a lot of resources, and starting by characterizing promoters. And here's just to illustrate how we use this particular tool. This is a gene, it's a transcription factor that specifically expressed in the G2 phase in the cell cycle, meaning it's just before it enters in mitosis. And well, Katerina did this characterization. He constructed for different reasons to a long version of the promoter and a short version of the promoter, and using the goose technique to show by blue coloration or histology, we know where the gene or the promoter is active. Here you have a lateral root formation, which is starting in the pericycle zone or near the pericycle zone of the root. It's forming a new lateral root, so we can just follow the root. And the description of the coloration that we have here corresponds to the part of the root or the new or the nascent root that is quietly proliferating. The same version more or less gives you the same description. And I don't know what happened here with these images. Anyway, you can see initial infections. Normally we have the nodal primordium, or I can use this. So this is very initial infection here, where the primordium is being formed in Phaseolus vulgaris. It's a very well-grown Phaseolus vulgaris nodule primordia and a well-developed primordia. So it is a promoter from a gene that is truly involved in cell division. Another gene that we use, it is the promoter of the gene P-null. We are not really interested in using genes or mutagenized genes because it's affecting the whole root. The whole root, I mean, the apical meristem in the root, it's a proliferative. So if we inhabilitate a gene, the cells won't proliferate properly. So we rather describe things and not necessarily inhibiting expression. So we just here using this promoter, we validate that that length promoter is working finely and lateral root is being formed here. You can just count that there are 4 layers that have been divided. The root is becoming a little bit at later stages, and here is the nodulation. But the most important was that when I was checking these roots at the microscope, there is an infection site here that you cannot appreciate because at that time I didn't have the rhizobia strain, but we can just count 4 small or shorter cells, meaning the cells are dividing there. So the project, the ELISA's project, when it started the PhD, was trying to figure out, it was a very difficult question. Are we able to detect when these particular cells starts division, having as a reference how long is the infection thread? It was a really difficult question. In terms of experiments, it was really difficult. She succeeded, and we summarize here the results. So since it was very important to have a diversity of molecular tools, we used the autofluorescence that is quite common at certain long wavelength in fluorescence to visualize the perimeter of particular cells. We have also developed or construct this turquoise NLS because we did have our interesting problem protein fused to GFP or YFP, and we already have GS red. So we need a third color, and we decided to have turquoise or turquesa. Okay? So probably I will ask Jesus for difficult projects to have the TNLS or protein. But Elizabeth, what she did a lot of confocal microscopy. Some of them during the pandemic, it was complicated, but she insisted that whenever she was watching the root there, in general, quite often, she found or she had the impression that the noodle was not always at the same position or in more or less the same position. Since these cells, this cortical cell here, is a cell that is large and has a huge vacuole, and the nuclei used to be on the perimeter of the cell because inside there is a huge vacuole. So she insisted that the nuclei sometimes was in the middle, sometimes here, the base of the cell, etc., etc. So I asked her, I forced her to count an incredible amount of images and looking for a description of where the nuclei are found. This is the nuclei. See, she counts more or less 80 nuclei here in an inoculated root. It's a part of the root in which she had to find root hair. So she was convinced that this is the cortical cells that are susceptible for infection thread. She counted the position of the nuclei, different positions here. He used as a reference the cells that are neighboring, or side flanking an infection site. So this is an infection site, and this is the distribution of the length cell that was there. She insisted that the nuclei was in the middle and the cells were becoming shorter. I mean, the cells, they don't become shorter, but the cells were shorter. So flanking this infection site, the cells have more or less the same size. And since we use this particular description, like stage 1, in which we have a microcolony, stage 2, in which we have a short or initial infection thread, it's advancing, but we are really careful that we were sure that the infection thread didn't cross to the next cell. This is the stage 3. And whenever we have seen that the infection thread had already invaded the next cell layer, we call that stage 4. So stage 1, the nuclei, the cells were a little bit shorter than this and becoming shorter and shorter as the infection was advancing. And then the position of the nuclei, we have 3 very randomly described, like at the base of this cell, the cortical cell, the subepidermal cell, at the middle, central part, or the apical, depending on where we found the nuclei. And it was striking, and I insisted, and she repeated the counting several times. It was quite striking when the root hair was at stage 1, having this particular description in which we cannot identify whether the membrane was already invaginated or not. So we call this stage 1. And the nuclei tends to be at the center of the cell. Later on, later stages, this rate changes a little bit, and you can have the basal and the base of the cell and the central side, okay, and so on. So what that was striking. So we thought that, considering what this is in the textbooks or several papers, which is not really well described, they used to say that when the nuclei is at the center of the cell, by definition, may start or may be primed to get divided. Happens more or less the same in animal cells. But in root cells, which have a huge vacuole, the presence of a nuclei in the central part, it's meaning something. And we thought, and we're still considering, that this is an indication that eventually the cells, subepidermal cells, we cannot distinguish whether they have initiated cell division or not. But since the nuclei is in the center of the cell, we conclude that, suggesting that the cell cycle has been activated. I remind you that these cells are differentiated, so soon they have to get divided. What is happening here? Is the genetical program that has to be adjusted? We don't have an idea. Several people have identified genes, but still it's an open question. So that's the main result taken from this particular slide. And then we use the cell wall of the fluorescence to have the description that we have already in terms of the length of the cell. But what was important is that I decided I wanted to see an indication that a real indication that the cell has been enter a cell cycle. And for me, it was the most simple thing is just to be sure that cytokinesis already took in place. And this slide, which is published by Elizabeth, summarizes what is well known happening in vegetal cells dividing. I must say that has been described in culture cells or in the apical meristem in the root in Arabidopsis. So we decided to take this as a real label, landmark, that the cell is dividing. Why? Because here we have an interface cell, which is, like regular cell. The position of the nuclei, not necessarily it's in the center of the root, but then it is known that when the cell goes to the G2 prophase in the cell cycle, the nuclei moves to the center of the cell, where prophase band, which is a particular arrangement of the microtubules, all the lanes that are here, like a yellowish purple, are microtubules that get organized here. It's quite well described since 30, 40 years ago. And this is the nucleus in the middle of there. And it's an indicative the G2 phase is happening here. It's by the end of that. This is preparing the cell for division. It is known that the position of that particular microtubule band, it's indicating the cell that this is going to be the dividing site, okay? So this is the plane that cells knows that it has to be divided. So the division progresses, and we have here the different phases. And in plant cells, there is particular characteristic. You have, the cell has to construct to build this machinery, which is called the phragmoplast, which has a particular goal to build from the center of the cell to the periphery a new membrane that has to be growing from the center to the periphery, which will become the plasma membrane and the cell wall that's going to divide these two cells. So I decided if we were able to see that particular membrane, which is called the cell plate, so we will be sure that the cell is being divided. I have no idea when it's going to be finished. And as a concept, whenever this particular cell plate reaches this membrane and fused to this membrane, that's post-cytokinetic cell. So Elizabeth used that protein, that gene, which is called NON, as already mentioned, and it's considered now like a cell plate molecular marker. Here you have immunofluorescence with specific anti-null antibody that you can just appreciate them. These dots are important because that particular protein, it is synthesized in the anaphase, which is half at the middle, let's say, of the mitosis. It doesn't start the cytokinesis by the end of the mitosis like animal cells. In plants, is more or less coordinated the middle of the anaphase. So all this is the characteristics I already mentioned. There is a particular marker. The gene is transcribed during the G2 phase. For that reason, with that particular characteristics in the promoter, we were able to identify the transcription factor that Kathy was working with. So it is transcribed during the G2 phase. It's translated during mitosis, anaphase I already mentioned, and is degraded post-cytokinesis. So it looked like it was a wonderful marker. That particular promoter contains a domain, a small domain, which is called MSA, mitosis-specific activator, that allow us to recognize several genes that are involved in cell division, plant cell division, okay? So using that particular marker, Liz took all these roots that have been infected with the rhizobia expressing the DS red I have already showed you. And she took dozens of images that were further analyzed. So the tricky thing is that she had to identify cells that had this yellowish label here. That's the chimeric protein that we can, Elisa constructed with the promoter that I show you that specific, contains the MSA fused to GFP, and by the end, the null. So whenever we see this yellowish or yellow bar here, means that the cell is dividing, okay? So this cell is not dividing. It has a microcolony and perhaps building a particular invagination or infection thread. That cell has, in stage 2, we can just see where is the infection thread elongating towards the base of the cell, okay? So that particular cell, since this is confocal microscopy, we do know that this is the cells that is underneath the infection thread. This is the subepidermal that is underneath, not any subepidermal cell, is underneath the infection cell site. So later on, when the infection thread is longer, trying to reach the base of that cell, the root hair cell. In here, you see there is a tiny, tiny branch of the infection thread. And the cytokinetic marker is indicating us that that particular cell that you have here is more or less finishing cytokinesis. The length of this cell plate is an indicative, more or less, the width of the particular cell. So this is really by the end of the cytokinesis. And whenever we check cells, epidermal cells that were already invaded by the infection thread, all of them were either very post-cytokinetic, or we didn't see that particular. Because because we cannot say that, if that particular cell finish mitosis, is going to degrade this label. And we have to wait till the next cell becomes with the cell plate formation. So with all this, we demonstrated that whenever in Phaseolus vulgaris, I would say that I didn't check other legume. In Phaseolus vulgaris, whenever we observe that the root hair, the infection thread exit from the root hair and crosses to the next cell, is already dividing. So we conclude here that even though it was a concept that the root hair studied in Medicago truncatula in Lotus japonicus exits the root hair, invades a cell that in Medicago truncatula and Lotus would never divide, considering this first layer. So in Phaseolus vulgaris, this coordination corresponds also to what we know now is the phase in which the cell is almost finishing division. We were really happy. We published the paper, and Elizabeth got her PhD, and we have still an open question. Because we can say that when this is happening, that cell is finished, is divided. When that cell started to get divided, it was just like a question that we wanted to address. So to address this, you have to know more or less in Lotus japonicus, it has been reported the timing between interaction between the bacteria and the apex of the root hair and the time when you appreciate a microcolony could spend 18-20 hours. So it is a long period. You cannot visualize when this is going to happen because you don't have a clue there. So we wanted to have clues there. Then the question was, in which part of the cell cycle is this particular cell? We know that it's dividing. Statistically, it's dividing at this stage. But we still have the question of what's going on, what turns or how or when the cell cycle turns on in that particular cell. So we were looking for specific markers that will tell us about the cell cycle phases. We have looked for several, and we work harder until we finish ending with the microtubules as an indicative that the cell has started a reorganization. The microtubules reorganize when the cells gets activated and goes through the cell cycle. In plants, more or less estimated in cell culture, will take around 24 hours. And we used to know that the interface here it is a long phase, but the G1 and the S is the most longer phase in the cell cycle that also applies to cell biology, plant cell biology. G2 is checking that everything is ready to get into mitosis. And mitosis is short. You have to be lucky to be a mitosis there. Whenever you see this particular thing, you know that it's more or less in the middle of the mitosis. So microtubules can label a non-dividing cells because it has particular cortical distribution. Whenever the cells start the G1 and to the S phase, turns to get organized. It is not as nice, this particular arrangement is rearranged. We can see small fragments of microtubules trying to get arranged. By the end of the G2 phase, you start to see that PP band that I already mentioned to you that determines the place, part of the cell that's going to be the division plane. If it's an asymmetric division, that particular band moves to the asymmetric cell division plane. But here in general, in the middle. And the cell cycle progresses, and the prophase is characterized by all these spindle characteristic distribution of the microtubules, more or less as in animal cells. So you see them in poles. Then the anaphase progresses. Here, support is supposed to start the building or the formation of the cell plate here, but the microtubules get arranged, forming the phragmoplast. And the phragmoplast is this arrangement. This is perpendicular to the cell plane, okay? And then you have the telophase is more or less, you know, the nuclear envelope is already reorganized, and you have two nuclei separated by the future membrane, and you have two cells. So we decided very naively, let's label the microtubules. And I say naively because it was not difficult to construct that marker. But the microscopy in a really thick root is really a task. But Eric is doing a very good job. So you have here images, and you can see the apex of the Phaseolus vulgaris root, which has not been described. Still, we don't know how many cells are in the quiescent center, which are supposed to be 2 or 4 cells. We'll soon respond that particular question, but not today with the topic. So you can see here, and I use this because it's a very proliferative zone of the root. The cells are dividing. Think that these cells are programmed to be divided. As soon as the first cell becomes divided, it becomes like a machinery building new cells. The most important is the first one, which is difficult to study. So these are quite proliferative, and we use that as a way to convince ourselves that the microtubules are working fine. So you hear the cortical distribution of the microtubules. It's not the membrane, it's the cortical distribution. And these 2 spots are representing that particular band, meaning you have an image there, and it's a 2D image, so you cannot see this in that particular plane, but you see the spots in which that image is indicating that in these cells, you have this part of the PP band that's arranging particularly microtubules. So we can just identify different stages in the cell cycle. So we just illustrate quite a few of them just to convince you that we have a very nice marker. So here you have the spindles separating to the poles. You have a phragmoplast here. So these are showing you these dividing cells. If we move through the root until we reach, or Eric, which was taking all these images, reach the elongation zone, meaning the cell becomes larger to get differentiated. The microtubules now is easier to see. You have the cortical cell organization, and you have this transversal because it's a plane. So you have these transversal microtubules here. This is quite a classic canonical image of non-dividing cell. But single thing, when we close up here, you can see this arrangement is completely different than this is the cortical one. The poles are being formed. So Eric has the poster 18 if you want to see what's going on in infection. Now I just spend a few minutes. We also try to use this spectacular marker that has been used in Arabidopsis thaliana, but it is true that when you check, the images are not as convincing as supposed to be. And in general, they present what's going on in the division zone of the root, but never present what's going on in the elongation and the phasation. Well, the most important is that we've decided, because this is supposed to be a marker that's telling you when the DNA is synthesized, the S phase. But this image is very nice from the paper, doesn't work that fine. So for the moment, we have blocked that particular characterization and we spend time in other markers. Just to show you here, this is the elongation zone, supposed to be that promoter, which is endogenous, is directing the transcription of that particular chimera. But it's supposed to be in a proliferative or dividing cells, but you have here an elongation cell. So we hesitate, although I must say that the nuclei, when you zoom the cells, it is interesting because it has a particular dot inside the nuclei, which is indicating that a particular cell is synthesizing DNA. So in perspective, still a lot of things we have to learn. We have many constructions still going on. So new students, very welcome if they are interested in trying to answer the question. A lot of images to be taken, analyzed, which is really a task. A lot of information that has to be integrated, a lot of things that have to be learned. But the most important thing is going back to our initial question. So how these markers are distributing, that is going to tell us how this rhizobia, when it's inoculating the roots, it's making changes that you can appreciate in the cells. So thank you so much. I already summarized the people, collaborators. Jesus, I think he wasn't very happy that I kept working Phaseolus vulgaris, because all these ideas that we had months ago, we haven't started. He has a very busy team, and we are quite busy now, so I didn't insist, but probably soon we are going to do some things. So thank you so much again to Jesus Montiel and Mario Serrano for this wonderful experience and the funding agencies. Eventually, thank you for your attention. If you have questions or students that they want to realize particular cloning and microscopy, very welcome. Send me an email and we'll be very pleased. Thank you so much.
We have now.
Thanks for your talk, Rosana.
Thank you.
My question is, your purpose is that cell division is essential?
It is essential.
Yeah?
It is essential. There are mutants in Medicago truncatula and lotus. There are mutants in which a particular gene is interfering with the cell division. And in general, the infection thread doesn't progress further. They doesn't exit the root hair and could become like collapsing in the root hair. But that's information that comes from mutants and from other models, and is considered like a classical.
Okay, but then, so what is that atomic size cycle?
There are also mutants that are unable to get infected and they never produce infection thread, but somehow they can just divide and form very weird proliferating nodule-like, but it is regulated. And there are some proteins that have been characterized that are necessary, and you can induce the formation of these irregular nodules without infection.
Does it think that the root affect cell cycle coordination?
Hasn't been described. Yeah, I guess that they know, and so I have no idea how the roots are, but I would say yes. But for that particular, I mean, for that reason, I think it was interesting to show and to have tools in which you have mutants that, especially kinases, that they are constitutively activated and showing that can they divide specifically there. And they are used to be called nodule specific, have no idea if they have a function in the rest of the root.
Here we have another question from Jose.
Yes,
Thank you, Rosana, and congratulations. Very nice work.
thank you.
Just a couple of questions. The first one is, how frequent is the formation of double nodules or triple nodules instead of a single one nodule? And what happens with the initiation process in this case?
Another notion that is very well accepted, that it is that, as Jesus already mentioned, you can just have a bunch of infection sites, a bunch of responses that root hairs will eventually be forming a root hair. But suddenly the root decides that if I am only supporting 20 nodules because it's too expensive to have a nodule, I don't care how many infection sites I have, I will develop only 20 nodules. That's the consensus. In Phaseolus vulgaris, the classical in wild type roots, what I have seen is that you can inoculate, and eventually the ground that is just beginning of the base of the root hair, nodulation is preferentially initiating there. So all the nodules start to develop. But since it's continuous, you can just go to longer part of the root, and you could eventually find primordia, etc. But it's quite difficult that the primordia will develop full functionally nodules when you have already a lot of nodules already function. And transgenic roots is just like transgenic roots. The rest of the plant is not transgenic. They don't follow our development program. So just I cannot insist that it's a correct, perfect, well-formed root, but it's a functional root and has a weird organization. It's weird. And we have already compared how many nodules we have when we have transgenic roots versus wild type, and they have less nodules for sure, and not as well distributed as in the wild type. But this is the toolkit that we have. And I think it happens in the same, in the other legumes as well, but I cannot say more than that. You can just have, like in transgenic, double nodules as well. So
my second question is, have you tried to disrupt mitosis by using antibiotics? So the hypothesis will be that you had mitosis in the meristems, in primary root meristems and lateral root meristems, but you also have nodules, and all these cell divisions processes in different regions are controlled by perhaps different factors.
Yeah, I would say that.
But you need mitosis to work.
Yes, for sure. So
if you use antibiotics, you attack the mitosis process. And what happens with every meristem and with nodules?
There is a drug that
is
tried. For that reason, yesterday I was saying, hello, you have to be sure that it's not affecting other processes. There is a drug which is called afidicoline. Afidicoline is described in every paper that is specific for eukaryotic DNA polymerases. So you eventually stop DNA synthesis. And it's the way in which, if you check papers in which they have a cell culture that has been synchronized, you can check, and they add afidicoline or afidicoline, whatever it is in English, and they stop. And all the cells are stopped in the DNA biosynthesis. They take off the drug and the cell continues. It's not dead. I mean, you have to control. So initially we say, very nicely, we are going to inoculate the root with rhizobia, and 10, 12, 24, 30 hours after, we are going to put some acetylcholine in the same site. And we'll stop division. I don't care the rest of the roots. We'll stop there. And I don't need transgenic roots for that question. Bad luck. Because I have been working with a lot of things, and I have been working with bacteria, and I don't trust definitions. So I say, if rhizobia is susceptible to acetylcholine, I'm going to be in a mess. So we did a growth curve. In the presence of acetylcholine at different concentrations affects bacteria division. Not at the rate that eventually will come important for studying bacteria, but for me, which is a very slow process, could affect. And it's mentioned in the literature that the colony has to be formed, meaning the bacteria has to be divided, has to be healthy to invade. So I didn't want to get in that mess, and I couldn't find another pharmacological treatment that will allow me to stop division and address the question. Just I know what is being described using mutants.
Maybe ceosine or bleomycin. I don't know.
We can talk later on and just eventually figure out.
Thank you.
I have tried or imagine that eventually can disrupt microtubule organization using orselin or these drugs that disrupts microtubule organization, but it seems tricky. So I cannot control the experiment. That's the point.
I think so. Thank you. Thank you very much.
Is there another question around? Well, if not, Mario, I think you. Who else?
Thank you.
I will.